on a kind of an extension project of of our main lab, the genetic engineering lab, where he is testing out different sugars, the effect of this different sugars, so sugars other than arabinose on the expression of the plasmid. All right, so other inducers, basically. All right, so without further ado, I would leave it to Anthony, and he will also address, I forgot to mention, I wanted to just give them a heads up, he will focus also on implementation, in addition to everything that we discussed. Thank you. Thanks so much. Thank you.
Well, thank you. I wish all my students do that every time I walk into class. I actually wish
all wish that.
I know. I'm going to order a room like this. This is quite impressive. I feel like I'm in a TED Talk, especially with this little speaker voice amplifier. All I need is a black coat to go with it, right? And in the darker room, so then I feel right in. So I'm kind of going to, so my purpose of my talk is to kind of take you back to why I got involved in this, what my experiences were, and also where it's led me just this past year, because I was selected to be part of the Master Teacher Fellowship Program, which is an additional program through ABE that allows teachers to get together and share ideas in science pedagogy, and also to develop a project. So it's something that it's all open to you, to all of you, because you're going to be soon in this program. That's why you're being trained. So something to think about, and I'll talk about a little bit more about that eventually. So I want to thank Shahira for inviting me, for being part of this group, and also for all the people who trained me in 2005. It's Wendy over there. Karen was there too, I think. I think maybe Joe might have been there too. So you're in great hands with these wonderful educators who, this program has just grown so tremendously that it's gone worldwide. And it started off here. So I'll talk a little bit about the origins of the program. So without further ado, so who am I? I know Shahira introduced you already, and it kind of reminds me of that joke by, if you're a Saturday Night Live fan and you saw the Phil Hartman, when Ross Perot, for those of you who can remember Ross Perot when he was running, he had a running mate called Admiral Stockdale, and he seemed a little bit befuddled. And there was a gag that Phil Hartman got on and said, who am I and what am I doing here? And sometimes I feel like that, especially when I go to class. So here I am. So, oh wait, that's the wrong movie. Oh, let me go back. So, you're probably wondering what that is. So that's me in my first year of teaching. So they had hired me to teach a class in archaeology. No, that's not true. That was Halloween. So back then when you start teaching, you're very enthusiastic and you always dress up. So I dressed up as you-know-who there. And so thought I'd have a little play with that. And so here's what I like to do on my free time. I like to play with fire. So what this is methane bubbles. So those of you who teach chemistry, actually even for bio you can do this. So you take a tray of water and put detergent in it, and then you pump methane into it, and then you get all these bubbles and throw match in it. It's actually part of a video that a student took of me, and this is a frame from it. So that gives you a bit of background on my years of teaching.
I like the safety first
I know,
cabinet in the back.
Am I wearing yeah, this guy.
Yeah, we're sketchy here.
You probably don't want to enter this science teacher's classroom after seeing this thing. So the kids always love this. They always ask me, can you do the methane bubbles? So you know that is. So here's my family. They're right here. This is, I think, in Sequoia National Park, right? Sequoia?
Yeah.
So this is about 4 or 5 years ago.
Yeah, about 5 years ago.
And there's my son Anthony and my wife Cindy, and they too are in education. My son teaches right now at Loyola High School, and he loves it. He teaches chemistry, and Cindy taught many years English now, and she's a retired school administrator. So you can see we kind of like the outdoors there, right? And then now here's my first AP Bio class. So I don't teach AP Bio more, but I taught it in 97. So I had a small class because I wasn't getting paid for this, but it was an illustrious class, especially the elderly gentleman there, who was my best student. He was so good. that he got a Nobel Prize for his knowledge. Can anyone guess who that is?
Francis Crick.
Yeah, that's Francis Crick. And I got actually to meet him.
Wow.
I took a workshop, and there was a flyer sent out that he was giving a talk at Cal State Northridge. And so I said to my students, there was only 4 of them, that young gentleman there is, actually he's only a year older than me now, this is years ago. He was also a co-teacher with me, so he said, we gotta go see him. This is a once-a-lifetime event. So we got into a van and drove down to Cal State Northridge, and he was giving a talk. At the time, he was working at the Salk Institute, I think, or down in San Diego, and he was doing work on vision, because he kept jumping. You know, Crick was always itching to do something new. So he was kind of an interesting speaker. Then we got to take pictures with him. So I think I told my students to say DNA or something, or maybe it was James Watson, I don't remember. So now on to my biotech experience. So I got involved this first with Jim Wolfe, and I don't know if you know Jim Wolfe.
Yes, I do. From College of the Cancer.
Yes, Jim Wolfe.
Yeah,
So
I used to teach there for a number of
oh,
years.
okay, so you know Jim
He was
well.
there back then, I don't know.
So Jim Wolfe, at the time I first met him, he taught at Cal Lu, and he was doing a workshop on these different biotech techniques, as you can see here. And this was at Pierce College. So, and that's the first time I actually had an introduction. I did very little in college with biotech, maybe micropipetting here and there and some gel degrees, but back in the 80s, we didn't do much of this in college. Now I think it's par for the course. So he walked us through these different labs and gave us these different modules that are student friendly, and so that's where I got started. We actually even did PCR, believe it or not. I actually did it with frying pans, and then we actually timed it, and we took the pocket peach tubes from this and that, and we had to time it. And it didn't work out, but it was just the experience that got the kids excited, so that was fun. So then what happened is I ended up taking a class with him at COC in biotech, for biotech certification. I got that word out. And so he continued his outreach program at COC, and then I said, hey, can I still borrow your equipment? And so I did. And the person who was responsible to maintaining and preparing the equipment was this young lady, Abby Bernstein, who was his tech, and now she went on to work at Amgen. And then she was also, now she's a senior advisor at Thermo Fisher, I think Wendy.
She moved back east, right?
She knows Wendy well.
I think she moved to the East Coast.
Yeah, she did. She's on the East, I think Massachusetts.
Yeah.
Yeah,
That's where
so
Thermo
you
is.
all know it. So Abby, so she was just quite, she's wonderful tech. She had everything just ready to go and prepared. And then she told me, she said, you know what, you like doing this, you need to expand your program a little more. You need to start doing these other things. Have you heard of the Amgen program? I said, no, that sounds great, Amgen, when you hear that's really cool. So she said, you got to get in touch with Wendy Johnston, and because she was involved in that. And so I said, so then I think either she contacted her or I did, and then I was told about this wonderful program that was having a training session in 2005 right out here at this wonderful campus. And so two of these faces should be familiar to you. And the gentleman right there is Marty Conda. Does Marty still come around to visit?
He's pretty busy taking care of grandkids now.
When he says he's very busy to ceramics. So these 3 wonderful people got me started, and we did a 5-day training, and just like you guys are doing here. I don't remember coming in this room. This is really nice. But it was wonderful. We had great lunches too. I think we had a lot of, there was a lot of gyros, wasn't there? A lot of, I think it was Daphne's, I think that was it, right? Yeah. So we always looked forward to those. And so with these sort of people, they got us started. So let me take a little bit back on the history. You can find this also on the website. These are, our founding fathers. So you've got, on the left is Hugh Nelson of Newbury Park High School, who I believe is retired, right?
Yes.
And then in the middle is Bruce Wallace, who's no longer with us sadly. But then Marty's there, and then this famous shot of them, this great shot of them. They actually started the whole program, and Wendy could tell you so many more stories about this, and probably Karen and Joe, because they actually Hugh knew Bruce well, and he would literally meet him at the gate, the fence, and then he would give him stuff. And then when Amgen got a hold of it, they said, "Uh-oh, that can't keep going on." And so I guess they had to kind of formalize the program and have their hand in the pie. But then I think Marty came on board late 90s, I think, more or less, somewhere around there. So they call it Bruce Wallace, but then now it's been since renamed, I think, for funding purposes, ABE. And then, so these two wonderful ladies here, this is the next leg of my journey here. So this is Gio, who was, I think, Shahir's predecessor, if I'm not mistaken.
Yeah.
She also was, she ran the site distribution in Pasadena. And so she told me, you know what, because I used to ask all these questions, you know, are you ever going to do a lab with this type? Are you ever going to do a license test? You know what, you have all these good questions. You really should try the Master Teacher Fellowship program, because then you could kind of meet other people who are thinking along your lines and start to develop projects. So I said, okay, I'll give it a try, I'll think about it. And then she nominated me, and then I told Elizabeth, Gio wants me to go. And she also is a ABE teacher. I got her involved in this. She said, no, you really should go, because it's a once-in-a-lifetime opportunity. It may not come again, you really should try it. And so, with the encouragement of both of these wonderful ladies, I said, okay, I'll give it a try. So I quickly had to put together a resume. I hadn't done a resume in 20, 25 years, so it's really good. It gets you up to speed on that. So I did. I thought, okay, maybe I'll get accepted, maybe I won't. And then I get a acceptance in late October. And so then I get to meet these three wonderful ladies again. And so they run the ABE educational aspect. They're out, and I think in Massachusetts, well, at least the two of them are. And so that's Jessica Julius, and she's also part of EDC, Education Development Center. And she is, I think she oversees the whole program. Is that correct?
That's Rebecca Lewis.
Rebecca Lewis.
Jessica's
so
just below her.
So, and then you've got Courtney Arthur, who's in charge of the MFT, director of the Master Teacher Fellowship. And then Dr. Candice Johnson, who is the technical advisor. So they kind of oversee this program, and we would meet with them through Zoom, because we have different people from different countries here. And so here's my cohort. So I've got two teachers are from Australia, two from the Netherlands, and one from Turkey, and the rest are from the US. I'm the one on the upper left, so you can see the age before beauty here. And so, so we got to meet, I met with these wonderful educators every month, starting December of last year, and we went up to June. And so we would just basically talk science pedagogy. Then they'd break us up into groups to talk about, you know, projects. And so at first, you know, we're kicking around ideas, and then one person, actually, Sydney Johnson, she had a cool project where she's developed a page gel with respect to the, because have you done the red chromatography purification yet?
This afternoon.
Okay, so she did a page gel goes with that. So that's just to give you an indication of what type of projects that some of these teachers do. And some of them are also working on lab exchange. Have you talked about lab exchange?
Oh, yeah.
Which is
Please,
cool.
Just a little time.
Oh, okay.
Every time they see me, I tell them about lab
Okay,
exchange.
so they're ready to throw tomatoes.
I'm sorry, can you explain what that is?
You probably, I know, you probably don't.
Deep in
Lab exchange, yeah, you probably are saying in your sleep at night.
I know.
Turn
Yeah, actually that is the term, sorry to interrupt, but I have to share with you all this. This is the term I use with all my students. When I want to get one of the major concepts across, I tell them this is one of those concepts where if I wake you up in the middle of your sleep, you're going to be able to tell me right away what that is. So by the end of the course, they have their head full of...
I know. I say the same thing in class. I go,
Yes.
if you forget anything, if you forget your mother or father's name or your own middle name, but you must remember this. So here's so this is actually a shot from one of my students' experiments. So you're doing this right now, the transformation lab. So you haven't seen your results yet. So it's kind of exciting. It's I have to say, I think it's my favorite one. Certainly one of my favorites, because it's so cool to work with bacteria, and then, you know, there's all the kids just love this lab too, because of all the different steps and all the different procedures involved. They really feel like scientists, because they have to wipe down the tables and follow proper sterile technique and so forth. So, and as you know, so you've got these three plates, and then the one on the left is the control, and you get a lot of bacteria because it hasn't been selected against. The one in the middle has ampicillin, and so you get these white colonies because it has the ampicillin resistant gene. but because there's no arabinose in that medium, they're not expressing the RFP protein, which the last one will, because that's where you get those nice red colonies, which is kind of cool. So when you put this in the incubator and then you wait overnight and then you're going, at first you don't see anything, and then a few hours later, next day you go, oh my gosh, look at this. The kids get all jazzed up. So it's pretty cool. And so you probably have seen pictures of this. This is the plasmid and all the different critical genes, the one for replication, and the RAC, which is important for controlling expression of the RFP gene. And then there's the RFP gene taken from, I think, sea anemone, right, which is discosum, I think. And then there's the PDI, which is the promoter, where the RNA polymerase attaches to. So this has been engineered to express this RFP gene. So it's been taken from the arabinose operon, if you know about the operons, which I'm sure you do. And so this is kind of my own little picture I put together. It's very rudimentary, but so I started doing research. So what happens with this lab, a lot of students ask me, or they wonder, okay, why is that red? What is so special about arabinose that turns on that gene? What makes it different? So we know that in the middle, the one with ampicillin resistance, there's no arabinose. The arabinose is lacking in that medium, so it's present in the last one. So how does it interact with the plasmid to turn on the RFP gene? So I did a little research, and what I found is that there's a loop in the DNA in the arabinose operon that doesn't allow the polymerase to get in there and start transcription. So there's these two proteins, the RSC proteins, remember that from the plasmid, they form this dimer, they kind of lock into place. And they, basically the gene, the DNA, the RFP can't be expressed because the DNA is kind of locked into place in this conformation. And so you have to unfold the DNA so that the gene can be expressed. So what causes that unfolding? So when you then add arabinose, the arabinose locks onto the arabinose C protein, and then it kind of unfolds and flattens out. But then I did other research, and I found out there's a second site called the cap site, like you have in the lactose operon, and it's called the catabolite activated protein site. And you need the CAP protein to lock onto that. And then if you have the CAMP, cyclic AMP, that also locks onto the CAP. And when you have that, then the whole thing runs smoothly. So you need those two molecules to play a role in the reaction. So you want the dimer to break up, then you want the cyclic AMP to lock onto the cap protein, which locks onto the cap site. Boy, it's all these chain of events here. And so I guess two events must occur for this expression. The arabinose must be present, and then cyclic AMP must be present. Now the thing with cyclic AMP is that if you have low levels of glucose, then cyclic AMP locks onto the CAP. But if you have high levels of glucose, then it doesn't lock on. So somehow high levels of glucose will shut off the whole operon. And I think the thinking is like black operon, if you have glucose present, then the bacteria prefer to use glucose because it's more energetically favorable than the lactose, right? So, so I started thinking about, well, okay, so what sugars could shut off this gene? So what could shut off the expression? So I started thinking about, what about glucose, sucrose, lactose, xylose? I even looked at IPTG, which is kind of this long name there. It actually was suggested to me by one of my colleagues in my cohort. So I started doing some research. I found that glucose is really the only one that does shut off high levels. And so, but I thought, let's try these other ones. And so I went ahead and then, set up these lab setups. So I, took the agar, and then, so this is basically an extension of the transformation lab. So we're going beyond that. So what you could do is, as an addendum or as an extension of this, go further and add and create more plates. So I put each plate has LB agar and has basically 1% arabinose, and of course the ampicillin. So the only difference is that each plate now will have an additional sugar. So there will be one with, glucose, lactose, sucrose, and any other sugar that you're willing to try. And then I added transformed, so I got a batch of transformed cells that were prepared, I think, for lab 6. So I added them in, and then this is my grid here. I know it might be, not too bad here. So basically the only difference for each setup is what the sugar solution is. So I did 2 concentrations. I did a 0.1% and a 1%. And then there's the rest of the grid. And you can check these later. Okay. Do you want me to go back or not? Did you get them all? Okay. And then this is my results of what I got. So I got all these, so for the 1% solutions, I just had these dense populations of red bacteria, which I expect, although I think that might be a piece of some mold growing there, if I'm not mistaken. So, as I expected, but then when I did the control, I got this. I got these white colonies with the glucose. The xylose is kind of pinkish, so I kind of marked that as red. And IPTG, I did get some white, but the other ones were all red. And so when I was doing this experiment, I started thinking, actually, I did this coincidentally because I went to Wendy and I said, I'm thinking about, adding different sugars to the setup. Will this work? And so she said, well, I don't know. We should try it. And so this will be fun in typical Wendy fashion, right? She's always excited. So, and then she called Marty too, and he said, well, I'm not sure, because he actually met with the researcher who had developed with Pico, if I'm not mistaken, right? There was someone who had, so he thought that he wasn't sure what would happen, so he said, let's give it a try. So Wendy then generously and graciously volunteered her lab and her students. These are the interns that work there in the summer. It's just a wonderful place, and these kids are just, you can see that they're future Nobel laureates because they're so good with their technique, and she trains them so well. They follow everything precisely. So there's a real purpose to the lab, and there's a real respect for precision and for protocol and for keeping sterile conditions. So just quite amazing. So then Wendy, I met with them and talked to them, and I gave her my grid, and she modified a little bit. So she talked to another doctor, another researcher there, Raj Pereira, who works, and he suggested that it might be tricky to autoclave some of these because you'll char the sugars. So she suggested, why don't you do one charred, one autoclaved? No, I'm sorry.
Filter sterile.
One autoclaved, one sterile filtered. Yes. Okay. And so that's what we did. And then we had also controls. So this is just the grid. It was more extensive, more plates. Do you want me to go back and get that? Or?
We're going to be provided with this IC.
Okay, that's fine. Okay. So here are the results. Now, we got more interesting results. So nothing was conclusive here, although there was one plate that just gave me only white colonies. And that was the one that the 1%, I think, autoclaved sugar. So I'll show you. So when you show me, here are her plates. You kind of see, this is just the control plates. You can see that there are a few colonies here, but they had actually done the whole lab from scratch to finish. So they actually started with, lab 2A, if I'm not mistaken.
Well, I have to just say that we used just a loop to make one streak.
Oh, okay.
So we did not put the 100 mics onto the, and spread, we didn't use spreaders.
Okay.
Hockey sticks. Yeah.
Yeah, that's what I did, and they didn't, so they just loop. And that's why you get fewer, but much more precise. So, and then here's the glucose results. So you can see how you get some red colonies. Although that one on the lower left has just kind of hard, two white colonies, and then some white colonies here. So it seems to me, it seems like glucose has an impact, but there are some red colonies in there too. And then here's sucrose, and you can see there's the colonies are a little small to see. There's,
Was this with a good batch of comp cells?
I'm sorry?
Was this with a good batch of comp cells? Because we
Yes,
had a bad
this
batch?
was-
Okay.
This was
Yeah.
the third time. We
Okay.
did it 3 times, and the kids poured their own plates. But the first time, we had an issue with the canned plasmid, and the second time, we had an issue with the competent cells. So third time was more charming.
yes, it's always a charm. So then there's a lactose, which again there's some. I think there's some red and white. So the windup was that there were some red colonies and white colonies in all the 3 setups. And so what are we to take away from this? You know, what is the conclusion here? Well, to me, it just means that I need to do more further study.
Yeah.
And,
Repeat more numbers.
But this is what science is about. Things don't always turn out like you want it to be, but I think that's the beauty of it. And so there are more questions than answers here. But then it's something I'm going to take back with my students, and we're going to further, study it. And then this, I did a write-up on this, and I submitted it to the MFTF group, and it should be up on the site. So I'm sure they walked you through this site, did they not?
Tomorrow we're going to do
tomorrow,
this
tomorrow,
more,
sorry.
yeah.
They'll walk you through it. There's all sorts of cool things here. There's Joe has some great, I think kitty paternity is fun, and the kids just love that. And the dog who ate the homework, right?
Yeah.
Those are great exercises, gelotophoresis. The kids always love those. But you can see how these are previous, lessons that were developed by previous teachers for previous cohorts. So there's all sorts of interesting things there that you can, check out and see if that fits with your, curriculum. And so here's my information if you need to contact me. My color doesn't look too good there. Huh? But my phone number and my email, and then I'm still at LaSalle College Prep. And then, I don't know if this will work. This is an iMove I made 9 years ago. Because I think I have to be connected to online, right? Yeah.
Yes, if you go.
The Wi-Fi is at the H? Is that the network? At the H? That's the guest network?
Yeah, it should be.
Okay, let's see.
So tomorrow when we go through the ABE website as well as the ABE-LA website, we'll show you where those teacher projects are. And one of the nice things about them is they are at different levels. So they weren't all high school AP biology teachers and AP bio level projects.
There we go. I love technology. Let's see. Let's play it for me.
You have to be on presenter mode.
Yeah.
You have to be on the show slideshow at the
Show
bottom.
slide, yeah.
Yeah, so it's
Yeah, it went dark. Just like in a classroom.
It does, yeah, I don't know, there is a delay,
No.
yeah, there we go. We have that too in the front, in the lab.
Well, it's on YouTube. If you hit Amgen LaSalle.
It should be, it might be in the background, and maybe that's what happens to it. Because if you get out of the slideshow,
Oh,
Escape? Escape?
I did.
Yeah.
Oh, is it on the screen here?
Something is not, okay, now what?
It does that, yeah. That's okay. Well, you can check it out at some point.
There,
Oh.
there we go. So it should be somewhere in the background. Oh, I think it's rebooting something, maybe?
Maybe.
Yeah. Can you see it in the
Oh, these ones
background?
I did buy.
Karen,
Betsy. Yes,
who's got the?
it's right here. Now's a good time?
Huh?
That's okay, sure.
Okay.
It's not, no, it's not.
Here.
Yes,
Okay, so-
yes, I get that, yeah.
So, as Mark Twain said, it's a terrible death to be talked to death, so I shall end it here. So, does anyone have any questions?
You have a veteran teacher here to ask all your questions. Take advantage.
I have a question about the interns.
Yes.
So they're your students that had gone through the ABE training with you, or ABE labs with you?
You mean the ones working with Wendy? No,
Wendy.
no, not these.
No.
No, these are
Oh,
Wendy's
okay,
interns.
these are Wendy's interns.
Although I have had some students in the past who have been trained by Wendy, so yeah, but not for this, not for this experience.
Okay, yeah.
There was a 10th grader, and the extent is one was the woman who will work tomorrow has a year with a post-bac certificate. So she's from Cal Poly Pomona.
Okay.
So we had a whole interesting team.
All right.
That was a great team, yeah. Yeah, a question over here.
Yes. So, what would you suggest moving forward? Is it going to be sterile filtered sugars or autoclave sugars?
We got growth in both. Although I think there was a concern about the stability of the sugars if you include them in the autoclaving process. So I think sterile filter is a way of assuring much better results. So I would probably go that route. Is that right?
Anusha is actually working for Raj.
Who's that?
Anusha is
Oh,
working for Raj.
Oh, you are. Oh, wonderful. Yeah. So I think, would you agree with that sterile filtration is probably the way to go, Wendy? Or?
It usually is in
Yeah.
media, yeah. You
We
usually
don't
sterile
have to do
filter
that with the
sugars.
arabinose, but
Arabinose is really.
we
So-
already had the bottles, little
Right.
bottles full. They did 100 mils of each of 12 different types. And we already had filled the bottles to go through the autoclave. We didn't want to waste what we had done, but we added the sterile filter as a new case for each concentration.
Yeah. So I think sterile filtration would probably go. Way to go. Any other questions?
I had a question.
Yes.
It appeared that one of the other sugars, the one with the long name that got abbreviated to the IP,
Yeah, the IPTG.
IPTG.
that also seemed to prevent the expression of RFP.
It did. At least when I worked it, although Dr. Pereira suggested that there's some concern with viability of cells when you use IPTG that maybe that won't work. But when I did mine, so I used transformed cells already, so it seemed to have shut down the expression, but then, I don't know, there's a question about viability.
So my question was, what would be the mechanism for that sugar to, do you think that sugar is impacting cyclic AMP concentrations inside of the bacteria, causing that, I forget what you said, the cap? Like,
So I think the IPTG is an isomer of allolactose, if I'm not mistaken.
Yes.
Yeah. So it's acting like a variation of lactose, but then the lactose didn't prevent the expression. So I'm not sure. That's a good question.
It seemed to work, and I don't know why, right?
Yeah. Yes.
Oh yeah, when you said you use transformed cells, are those, did you get them from like an LB amp plate or from the LB amp?
I got them from, so the sites already have, when you do lab 6 for the chromatography, there's 2 ways you can go. You can actually take some from lab 5 or you can get them already transformed. And so that's what I used.
My question is if they've been exposed to arabinose before or if they've,
No, they
you
have
know that
not.
they're transformed but they haven't been.
Right,
Okay.
Yeah.
because I made them. So if you had started and transformed yourself, then you would have had the LB plate, and well, it's a lawn there, but you could have seen whether you had reduced growth with the ITPG.
Right,
Yeah, but
yeah.
you just did the LB.
Yeah, so I went
The
to
LB.
Yeah.
And just to clarify, the purpose of the experiment was to determine whether or not by adding the sugar you could not affect growth, but if you
Right.
could affect expression of
Correct. Yeah.
the protein by preventing its transcription. And so you would only be able to see if it worked if you gave it a Ravenous and
Yes.
the sugar.
Because my concern is there another mechanism going on that prevents the expression? So you need a Ravenous to have the gene expressed, but if there's another sugar present, will that prevent the expression even though Ravenous is present? So there's some type of, other switch going on here. So that's my concern. So it's still something I think that needs to be studied further. I think someone did an experiment, found somewhere where they did it with P-Glow, and if you add glucose and you have a Ravenous with P-Glow, then it shuts down the expression of the P-Glow, the Biorad P-Glow. So
And
I saw
is
things
that where
like
you
that.
determine the concentrations, the 0.1% and the 1%?
I got, I figured, I figured I want to use, I think the Ravenous is about 1.1%. Is that roughly correct?
Yeah, it's like 1 gram per liter, so 0.1%.
Yeah, so
Yeah.
I figured I wanted to use the same concentration of Ravenous.
Mm.
I did 2 trials of this, so when I first did it, they were all red. And then when I upped the concentration, I go, okay, maybe the concentration has to be increased. So when I did that 10 times more, I noticed that's where I saw differences. And so this begs the question, maybe you could do other different experiments where you vary the concentrations. And I think I'd focus more on glucose in the future and see varying the concentration, see at what point does it express, at what point it doesn't, and take it from there. But I think what it shows here is that you can take any of these labs and then start thinking about how you can go beyond these labs and what extensions you can, apply to these labs. And then I think that's where some of these teachers were thinking along too, like Sidney Johnson up north, who wanted to do a page gel with protein chromatography. Someone had a question? Yes.
So, I'm a bit confused. When the arabinose sugar finished, the gene will not be expressed anymore, so the colonies will show white, like around the edges,
Yes.
right? And so you are mixing the other sugar and together. So how do you differentiate that? Now it's not expressing because of arabinose sugar ran out, or because the other sugar was in the water?
Right. So at that
How do
point,
you differentiate there?
Oh, wait, I'm sorry. So
Yeah.
Kimmy, are you talking about satellite colonies because of ampicillin?
No, just the running out of the arabinose sugar.
Once it's all consumed.
But I think the
It's
thing is that
all-
we were saying is because of the transformed plasmid that there isn't any enzyme to break down arabinose, so arabinose will not actually
Right,
decrease.
it's not metabolized. And
That's
as
what
the
Karen was
cells
saying the other
grow
day.
and die, they
Uh-huh.
release arabinose into the media or onto the plate.
Oh, okay.
But the glucose actually would get consumed.
Right.
Right.
It get metabolized,
Oh,
right.
I see.
Because it's actually preferential.
Right.
For bacteria to take over.
yes.
Really, thank you for your presentation
you're
and seeing that there's teacher developed materials and seeing you go beyond what was given to us is very inspirational. And one of the things I would like eventually for even my own students is I'm always trying to encourage them to take what we've done and hopefully encourage them to go beyond that. And is there a pathway or a more concrete way to use that kind of encouragement and come up with a project or experiment beyond what we've done? Maybe it doesn't even have to be from this particular experiment, but something that maybe Amgen can support. Do they have, maybe some kind of list of things that, problems that need to be solved, and then
Yeah. So
As an
solve it.
extension project,
Yeah.
yeah.
We did actually have some interns in Massachusetts working on some problems that we had that the master lab technicians didn't have time to work on.
Sure,
So
yeah.
Ollie worked with some interns back east to solve a problem with us. Those were undergrads. And then we will try and support you even with materials. And, as teachers start up getting equipment, we go to the warehouse, we can even get things for you.
We can
treat
find
your
ways to
coordinator
get them.
nice, we'll treat you nice.
I mean,
One of
the dream is,
Yeah.
I would want one of my students to win Westinghouse. It's not called Westinghouse anymore, but
Yes.
like something like that, where
Yeah.
they're like, oh, they've
That was
understood all the concepts and they said, well, why don't we do this? And
I know
to
that
be able to give them
in our
that
discussion,
opportunity.
when we met monthly, it wasn't just about working on our project, it was also about science pedagogy and scientific inquiry. So a big concern now, and if you teach AP Bio or maybe AP Chem or any AP science classes, they want to guide students more in an inquiry-based lab and have them develop maybe their own protocols or maybe extension protocols. So I think if you open it up, maybe sometimes some teachers will create like a section in their curriculum where they have a project-based learning and go, okay, here's what we've done. Do you guys have any ideas of what you'd like to do? Or is there one of the labs that you'd like? Is there a possible extension that you'd like to explore on your own? And sometimes those types of ideas can create the seed that students will run with and then blossom. So I don't know if there's any easy way to that, but it's just kind of like basically, I remember hearing one time at a workshop, you could take any lab that's already canned, and then if you change it tweak it so that it says, well, what would happen if you do this? So you could convert these labs into more student inquiry, I guess, and then that opens up more the door for projects. And then you find that the more you tell students, like, the more labs that you give them that's already at least canned labs, maybe that's too strong a word, but labs that have everything spelled out. And then they can go through it, and you can even repeat them. But I find that there's less thinking involved. Whereas when they start developing their own protocols, then they start thinking about what if I change this variable, what if I change that, and the more you can guide your students in that direction, that will encourage more creative thinking, I think.
Right, so I would like to concur, because I think the students have to do the CAN lab once, because they have to learn the technique. And then afterwards, then you can start talking about extensions. But I feel like the more teacher gatherings you come together and you talk to other teachers, because we used to have we used to have an annual teachers meeting, and we would have a breakout where the veteran teachers would talk about different things that they had tried, and that's where you hear a lot of things. So if that's something you're interested in, like today, I can give you some of the different extensions I did with the chromatography lab, or what other teachers have tried. So yeah, so I think, just sharing of ideas, I think that's important. Sometimes I think just going through the exercise of what's another thing that you could change, even if you don't actually do the lab, but going through the thinking process and writing out the protocols and what is your control, what is your experimental value, I think that's really important for the students. Yeah.
So yeah, and also to concur with that, the teachers who are part of that program, the master teacher, they have their own projects, and so they have these projects usually listed, I think, on the websites of those of the program.
Yes.
So whatever.
Yes,
so if you
we'll
go, so you will see that tomorrow if you go to the ABE websites of the programs worldwide, outside LA. You're going to see some of these projects posted, so you get ideas, and then we have the annual teacher meeting in September. So
The reason why I bring this up is I also teach PLTW, and the 4th year is biomedical innovation, and it's very unstructured. I could literally have them just do an independent project. If the equipment is there, if I have the equipment,
why not?
then why not? Yeah, exactly. Why not? Why not? So
We've had classes where, I've had students even do individual projects, but the class decided they wanted to test what happens when you change the heat shock time.
Yes.
What happens when you change the heat shock temperature? And I'm telling you, a couple dozen plates from us is nothing, so we're happy to help you to do these extension labs.
I just wanted to add, my high school teacher, in addition to the ABE labs afterwards, we got to go to Channel Islands for a week and do DNA barcoding and see the different diversity of bees and bats and fish. And so we had collaborations with universities, and we were able to intern and do research with these universities. So CSUN was one of them, Channel Islands was another one.
Wasn't that barcoding? Was CSUN?
No, that was with
bees?
Channel Islands.
Oh,
So
okay.
they went out to Channel Islands for a week and did all that DNA barcoding.
And you can also do, so to cover that, Dolan DNA Learning Lab also has DNA barcoding.
Dolan.
Dolan.
DNA.
The Dolan Center,
Uh-huh.
the
at Cold Spring Harbors. Yeah. So you have to attend their teacher workshop. I just held it at Santiago in Orange County. And once you're trained, then you can get access to those things. So yeah, I think the more you talk to other teachers and find out, that you'll get plenty of ideas. And then once you have your own idea, you could do just like Anthony did, apply for the Master Teacher Fellow. And then that gave you a little bit of extra time, or did it buy time for you? Or?
They gave you a stipend.
There's a stipend. It's a $2,000 stipend, and there's another grant they're opening up another grant to pursue what we've initiated. So here's a stipend, and then what you do is you meet once a month through the Zoom, through video conferencing, and then you discuss. They break up. So we discuss our projects, but then a good part of it is given to discussing science in the classroom. How do you approach it? What best practices? What do you do? What keeps the kids enthused and energized? And I know that Jessica Julius said she's very big on that, on forming these partnerships with communities and schools and trying to find the spark that ignites this interest in science. And you probably have noticed that maybe in your classes when you're doing a lab, and you always get those kids, sometimes they're the kids who could be a bit of a pain, but they always, what if I tried this? What if I tried that? And if they do something within reason and is safe, and you go, okay, let's try that, and then ask them to write it up, then somehow that gets them going. And sometimes you get kids who are maybe not the best students, but that could be an incentive for them to get hooked into science. And I found sometimes those kids, and have you ever noticed that sometimes some of your best kids with lab technique is not always the best students with testing, and sometimes it's almost like and I've seen kids who all of a sudden they just shine in the lab because they're good at that, and then when it comes to tests they're okay. But I noticed that they enjoy the kinesthetic aspect of science and they really get involved. And I think it's things like that. And I'm always looking for new ways to also to keep that interest going. It's an ongoing thing. Teaching, you can never be a perfect teacher. It's a profession where there's no one's perfect, and you have to keep growing. I mean, we need several lifetimes to get to that, right? So that answers.
Well, I want to make where is it? Oh, it's up there. All right. To that point, we have a wonderful young woman who worked very hard on your project. And would you like to come up front? So I said that we had between a 10th grader, this young man Max, and we have somebody who had post-bac classes. This is Lauren. She's going into the 11th grade at the High School for the Arts at Cal State LA. This is her second summer with PBC, and this is her last day this summer because she's getting on a flight tonight for Taiwan and vacation, and so it's appropriate to give you your certificate, my dear.